we're all very excited after that initial session, and looking forward to an opportunity to discuss in the roundtable some of the enormous number of questions that have come through from everyone. Thank you, everyone, so much. Perhaps, Tony, if you want to take this one first. What is the greatest challenge for AI currently to overcome?
As I indicated in my brief video section, and one can imagine applying AI/ML to a number of different areas, but I'm most excited about applying it to those areas where the nature of the problem and the evolution of the technologies that allows us to generate the data flows that are so important in getting really effective AI/ML align in an important way. And for me, that is this both fascinating and vexing problem of the relationship between genetic variation and traits and disease outcomes and the inspiration for new medicines. And others on the call can speak with far more authority to the details underlying that. But fundamentally, we are going to see a revolution in genetics and genetic medicine in the coming years. And I'm very optimistic that AI/ML is going to help us find a way through, first of all, being able to manage the huge volumes of data that come from modern techniques such as images or highly dimensional omic measurements. But even more excitingly, because of the sort of work and Mihaela's focus, for example, on creating white box solutions from black box methodology, that we might find fundamentally new things about biology. And so I've been in the industry now for almost 30 years, Andres, and I'm a drug discoverer by training, and I can't think of a more exciting time. And I'm so pleased to have this opportunity to be working with you all on what I'm hoping will be a new horizon in our approach to drug discovery and development, but fundamentally by helping us to choose the right targets in the first instance. We've led publications on this, demonstrating that when you look at drug discovery and development programs in their clinical phases, those programs that are lucky enough to have what we might call highly credentialed genetic evidence enjoy about a twofold improvement in their success rates through those phases. And for those of you who haven't looked at the industry in detail, you might think, oh, twofold doesn't really add up to that much. But in an industry in which 95% of what we do fails, and does so over typically a 15 to 20 year period, improving that 5 or 2% success rate to 10% is going to have a massive impact for patients.
Great, thanks, Tony. Well, I share your excitement as well. So the same question to Jim then. So the greatest challenge for AI?
Yeah, thanks, Andres. So I'm going to take a slightly different perspective, actually, because I think Tony covered the sort of scientific elements quite well. I think there's a huge human challenge in terms of successful implementation of AI, in that any advanced technology only really works if people embrace it and adopt it and actually use it to improve the way they work, and in this case, improve people's health and improve the development of medicines. So that's why I think it was really critical that as we started to put the center together, there's components in there like ability to create prototypes and demonstrators, a laser focus on focusing on real world problems in healthcare and medicine. So it's not kind of, academic research over here, it's very much plugged into the problem space. And so hopefully the biggest challenge, the solution to that challenge is something we've already baked into the center because of the partners we've got and because of the mindset we've got. But I think that's really critical. And we can see also that unfortunately, it's often the Achilles heel of a technology if that wider conversation with the public is not conducted and is not done early enough. But I feel confident with the centre that because of the work that I've seen previously from Mihaela and other groups at Cambridge around ethics, for instance, it's a big hot topic at the moment. How do you do ethical AI? How does this really incorporate in people's lives when you're making big decisions? And you might argue there's no more decision than making decisions about your health. So I think we need to keep a close eye on that and make sure that we're doing great science and we're taking care of the human side as well, so that the great science we develop with AI gets successfully implemented and makes a real difference.
Great, thanks, Jim. Mihaela.
So thank you very much, Tony and Jim, for joining us and for the support for our center. I also have a little bit different question than Andres to both of you. So it's clear that clinical trials are somewhat at the forefront of really adopting many of the drugs that you are developing. So in that context, there are many challenges that we could address in clinical trials using machine learning potentially. But whether it's with machine learning or more generally, I wanted to ask both of you, which one of these challenges you feel that is the hardest and where there will be the most gain from a new approach, potentially using machine learning?
Okay, well, I guess we could go the other way around in this one if I start. It's always hard to pick one thing, isn't it? What is the hardest challenge? I think, as you and I know, Mihaela, there's loads of places we can look at interesting projects and solve problems with AI. But I think a really interesting area to look at, which is a tough nut to crack, is how you sort of do an ongoing accumulation of evidence. So one of the silver linings we've seen from the last year, for instance, is that it is possible to accelerate the way we look at and analyze data from clinical trials and use that to make critical medical decisions in a crisis. We don't always want to be in crisis mode, of course. So during other times, how do we gradually accumulate evidence and use smart AI to look at that to say, you know, when should this medicine be put out there, made available to more people? How do we track its use, its effectiveness, its risks, etc.? That really is a hard problem, because I think it involves transitioning from a paradigm of very discrete experiments today, mostly discrete experiments in clinical development, to a much more continuous evidence generation. And I think we'll need computational and ML related help to do that. I don't know exactly how to solve it, but as I say, we've shown that we can speed things up and do them more continuously when we're really pushed to. So how can we take that forward with us into the future?
Thank you, Jim. I don't know, Tony, whether you have anything to guide us with in terms of clinical trials. You are on mute, I think.
Let me offer a build on what Jim said. And I have an old colleague who used to describe to me that there were fundamentally two problems associated with the discovery and development of medicines. And they were, first of all, the design, the discovery of the medicine itself, and then secondarily, showing that it actually works in a patient population. And the diseases that we are focused on now that have huge potential societal impact have characteristics that mean that we absolutely need to be able to do a better job of pinpointing patients and then being able to, if you like, extrapolate from early events into ultimate success. Otherwise, we find ourselves in a position in which the cost of demonstrating the effectiveness of a medicine, and one in which we are quite rightly challenged with showing that those medicines deliver substantial improvements over existing standards of care, becomes increasingly problematic for the industry and for the discovery and the continued discovery of new medicines. And so I'm particularly interested in the work that you described during your presentation associated with pinpointing patient populations and being able to extrapolate longitudinal effects. And of course, to pick up on some of the other aspects that we've described, the growth of our understanding of biomarkers and in particular, the ability to use the advantages that this type of data analysis might bring to extrapolate from readily obtainable peripheral biomarkers into tissue dysfunction that is just so much harder to access by other terms. So I've deliberately gone away from genetics and target identification. There is a very, I think, clear connection between the two things that I've just described, though, as well, which I'll highlight. that of ultimately, in the cases where we can enjoy a, if not precise, a clear relationship between phenotype from clinic to laboratory, then we're also increasing our chances of having confidence that we've discovered medicines that will work, and the more precisely we can describe phenotype, the more precisely we can identify the particular patients who will benefit from that medicine.
Great, thank you, Tony and Jim beforehand. So apologies if we don't get to your questions during the live session. We're going to break Zoom with a number of questions coming through. So thank you very much for your questions. Keep coming them in. A question for Mihaela then. Could you please shed some light on the generalizability of machine learning models, and also during the model training process, how can this be assessed? Access to healthcare data is not easy, so while observational data may suffer from sampling biases. So I guess there's a couple of questions.
Yeah, there are multiple interesting questions related to that. So clearly with machine learning, we want to make sure that whatever model we build on a particular population, we understand when it's transferable to another population. And there are multiple things there. First, we would like to build uncertainty estimates, and our center and our group in particular has done quite some work in recent times trying to understand whether we can provide hard confidence guarantees associated with the transferability of a model from one domain or from one population in one particular country to another. And what we would like to offer is confidence estimates associated with these different types of predictions. So we don't want to just issue predictions, but also to understand when these predictions are trustworthy and for what types of patients and classes of patients. And again, this is challenging by itself because often in different countries, so we often look at populations in United States and the UK, United States and Asia, etc. And often there is not only difference in the type of population, the comorbidities, the age groups, the genetic factors, but even the standards of care are different. And what types of variables are collected may be different. So we have distributional mismatches and features mismatches. So this represents also an important clinical problem, but also an important machine learning problem. So work in transfer learning, out of distribution detection, represent important frontiers where our center is doing a lot of research, both my group, Jose Miguel's group, as well as Pietro Leo's group, all of them trying to look in this area, and more will be done. Now, going to the question of the availability of data, which I think it's really very important. Getting high quality data such that this research can happen is key. Without data, we cannot do machine learning. And for that, it is often complicated to obtain new data sets for machine learning researchers, as well as provide them to others to verify the research we are doing. So an important related frontier that we are engaging with is that of generating synthetic data with differential privacy guarantees. So the focus in that research, and this is a machine learning research problem, but it's very pertinent to healthcare, is to take data sets that are private and generate synthetic data with differential privacy or other types of privacy guarantees such as GDPR guarantees, and then they make them publicly available such that others outside the center or outside the NHS organization or out of other organizations we may collaborate with can also have access to this data, check our models, build their own models such that we can all become better. So synthetic data generation represents also an important frontier on providing data sets and making data sets available. So that's the answer to the second part of the question.
Thanks, Mihaela.
Andres, maybe I can reciprocate asking you a question. So one of the questions that was asked in the chat was, what exactly are these protein activity graphs describing? And are they based on physical interactions?
Great question. Thank you, Mihaela. So not necessarily. So it's an opportunity to encode all existing biological knowledge about protein-protein interactions or signaling pathways. So at one extreme, you can look at proteins that are co-expressed but more often we want to curate those so that we're dealing with proteins that have biological interactions, which we can then model as part of the ML framework. I think that was quick. Yeah, great. So Mihaela, sorry, I'm just struggling to, because there's so many coming through now. So another question, perhaps related to your, no, here, another question. How does the centre think about the imbalance of certain socio-demographic groups involved in the data that may associate with bias in the model prediction? It's a great question.
Yeah, so it is extremely indeed important, as I mentioned a little bit in the previous question as well. So we have different types of populations, potentially different types of biomarkers that may be learned. And what we need to learn is what is a bias and what is variation due to the practice of medicine, and what is variation because of the unique characteristics of the patient. So I often say we do want variation that is specific to the patient, such that we can indeed practice precision medicine or bespoke medicine. So we want to make sure that indeed, if a particular type of intervention or risk is pertinent to the individual, we do uncover that. But what we do not want to perpetuate and replicate is biases or variations that exist due to inequalities in treating a class of patients. So for that, an important branch of our research is to understand human decision making as well, and understand whether machine learning can help us understand these biases and remove these biases, such that we can indeed, using machine learning, lift all boats. So in countries, for instance, such as mine, where there is a lot more inequality in the delivery of care, how can we in such countries use machine learning to improve the healthcare and make it more consistent, independent on where there is delivered? So again, I think machine learning is a very critical ingredient there. On one hand, providing personalized, bespoke medicine that is pertinent to the individual and is best optimized for the individual at hand, but while being mindful that we want to remove variation due to practice and due to potential biases.
Great, thank you, Mihaela. I will ask you another question, which is, I guess, an opportunity for us to promote the centre. So what are the future plans for the Cambridge Centre for AI in Medicine in recruitment of staff and students?
Oh, that's well, that's a question that we should both take. So thank you for this question. So as we speak, we have the center has several positions for PhD students. I think that this is the most important part to communicate. We are looking for bright PhD students that come from a variety of domains. As you can see, the core team is very diverse, contains machine learning researchers, health data scientists, clinicians, management, variety, very different scientists like Schaar. So we have a very diverse core team, and more people from the university will be engaged with us as well. So we encourage a broad set of students to apply for these positions. As I mentioned before, we are looking at the international talent. We hope that many students from all over the world will apply for these positions. And the unique asset, I would say, of the center is because it is very collaborative between ourselves and also with pharma, we will make sure that we will have very extensive set of mentorship for the students. So unlike in a conventional lab where maybe you would have the advice of your own advisor only, here there will be a shared mentorship program across many individuals. So that's a unique opportunity. We also have at the staff level, we have 2 positions that are actually going to be closing very soon. So we really hope to recruit 2 staff members, both for the communications position in our lab, in our center, as well as for a center coordinator. And I hope we are going to be able to recruit some very talented individuals there as well.
Good.
Yeah. So Andres, maybe one question to you that's somewhat related, but a little bit different. A question came as to, will the center have a role in engaging and educating future and current doctors for the challenges and opportunities ahead?
So I absolutely yes. So it is one of our missions is to engage with the clinical teams two of the faculty members are clinicians embedded in and driven to implement AI solutions to clinical problems. So we're very excited about the opportunity of engaging with medical students, preparing them for the new future of medicine, which will involve AI and machine learning. So definitely.
And if I may add, Andres, we also plan to have with the center many different tutorials that we will be providing together with our pharma partner, but also just by ourselves. So please engage with us in the various tutorials that we are going to provide. We are going to have multiple mini conferences throughout the year and seminar sessions. So these are all activities aimed at educating as well as engaging the broader community, not only in Cambridge, but all over the world. So we hope that you will stay tuned and engage with us.
Brilliant.
And maybe on that note, I don't know whether you have another question, Andres?
No, I had a great. There was one very good question, which is: are there plans to combine biological multiomic models and drug synthesis that Andreas mentioned into clairvoyance?
Yeah, so I guess this is exactly what we are trying to do, to bring together the clinical and the omics world together. And what is nice about clairvoyance, which is this package that Ari Ercole from our center and our group has built so far, is that we want to make it open source and broadly extensible. Right now, it's focused on the clinical area and more on hospital care, but we hope to extend this to out-of-hospital areas and not only include clinical information, but the entire biological information that's available about the patients as well. So thereby using resources such as and capitalizing on resources from UK Biobank, as well as many other collaborations we plan to have with NHS.
Yeah, brilliant.
And maybe I want to thank again to Tony and Jim for joining us for this session. I hope they will continue to follow this event today. And especially it's my pleasure now to move to the next stage of the session today and introduce several talks from our brilliant PhD students from multiple research labs. We hope that you see the type of research we plan to do, and that many students who may follow this engagement today are going to be excited by this and reach out and subscribe for our PhD positions.